So part 2, would autonomic postganglionic fibers have a faster or slower conduction speeds than somatic motor fibers? What would you say? Well, how does myelin affect conduction speeds? If this is a somatic motor neuron, and this is a pathway of the autonomic motor neuron, how does myelin affect conduction speeds? In the presence of myelin, conduction speeds are increased, right? So if the nerve fiber, the postganglionic fiber here, is unmyelinated, nerve conduction is going to be slower in the case of the autonomic postganglionic fibers. Preganglionic versus postganglionic motor neurons in the autonomic nervous system. The preganglionic neuron is the first of 2 autonomic motor neurons. And the cell body is found either in the brain or the spinal cord. This preganglionic neuron always has a myelinated axon, the preganglionic fiber, that passes out of the central nervous system as part of the cranial nerve or spinal nerve. And it extends to an autonomic ganglion and synapses there with a postganglionic neuron, which is unmyelinated. The postganglionic neuron is the second of the 2 autonomic motor neurons. And in contrast to the preganglionic neuron, it lies entirely outside of the central nervous system. Its cell bodies and dendrites are located in the autonomic ganglion, and it synapses and is capable of synapsing with 1 or more preganglionic fibers here. The axon is unmyelinated, and the axon of the postganglionic fiber will terminate with the visceral effectors. Remember what the visceral effectors are in the autonomic nervous system: smooth muscle, cardiac muscle, or glands. Oftentimes, the sympathetic nervous system is called the thoracolumbar division because the fibers exit the spinal cord by way of the thoracic nerve 1 to the lumbar nerve 2. From T1 through T12 to L1 and L2 are where the nerve fibers for the sympathetic nervous system are going to exit the spinal cord. The cell bodies of these neurons are found in the lateral gray horns. So the cell bodies of the preganglionic neurons is found in the lateral gray horns. If you remember, the lateral gray horns in a cross-section of the spinal cord are only observed from T1 to L2. And they contain the cell bodies of the sympathetic nervous system. The preganglionic neurons of the sympathetic nervous system are going to lead to the nearby sympathetic chain ganglion or paravertebral ganglion. and then the sympathetic nerve fibers can be distributed to every level of the body. So nerve fibers of the sympathetic nervous system only are going to exit from T1, the spinal nerve T1 through T12 to L1 and L2. This divergence, this ability of the sympathetic nerve fibers to be distributed to every level of the body demonstrates the high neural divergence. So the nerves are only coming off T1 through L2, how nevertheless from the sympathetic chain ganglion, which is right next to the vertebral column, these neurons can then, the postganglionic fibers can then be distributed to every level of the body. So high neural divergence. The two-neuron pathway of the sympathetic nervous system is short preganglionic fibers because they only have to reach to the paravertebral ganglion or the sympathetic chain ganglion, and then longer postganglionic fibers that go all the way out to the effector organs. This paravertebral ganglion or sympathetic chain ganglion is connected to the spinal nerves T1 through L2 via two branches. The branches of the rami communicante. Remember when we talked about the spinal cord, chapter 13, we talked about the rami communicante. So these are involved with the sympathetic autonomic nervous system. The preganglionic fibers are small myelinated fibers that travel to from the spinal nerve to the sympathetic chain ganglion by way of a white communicating ramus. That's white meaning it's myelinated. You remember the white versus gray matter, white meaning myelinated. Postganglionic fibers leave the sympathetic chain ganglion by way of the gray communicating ramus that's unmyelinated. And these postganglionic fibers extend all the rest of the way to the target organ. So white communicating rami, the yellow here, white communicating rami carry preganglionic sympathetic fibers from the spinal nerves to the sympathetic ganglia. The gray rami communicantes carry postganglionic sympathetic fibers from the sympathetic chain ganglia to the spinal nerves. Gray communicante. After entering the sympathetic chain ganglia, the preganglionic fibers follow one of three courses. they can enter the ganglia and synapse immediately with a postganglionic fiber. Enter and synapse immediately, which then ultimately will lead out to sweat glands, piloerector muscles, which are involved in formation of goosebumps, blood vessels, skin, and skeletal muscles. Another route that these preganglionic fibers can follow is to travel up and down the chain ganglia and synapse in with postganglionic fibers at other levels along the chain. And then those postganglionic fibers go out to internal organs, such as the iris, the salivary glands, the lung, the thoracic blood vessels, and the esophagus. The third route that these preganglionic fibers can follow is to pass through the chain without synapsing with a postganglionic fiber in the chain and continue as splanchnic nerves that lead to collateral ganglion and synapse to postganglionic fibers in the collateral ganglia. And the target organs of these preganglionic neurons are going to be to liver, spleen, adrenal glands, stomach, intestines, kidneys, urinary bladder, reproductive organs. The collateral ganglia, or prevertebral ganglia, contribute to a network of postganglionic neurons called the abdominal aortic plexus. This plexus wraps around the abdominal aorta. Most postganglionic fibers will exiting this plexus will accompany arteries of the same names, with the same names as the nerves, and follow the same routes to target organs. So, preganglionic fibers from T5 to T9 or 10 are going to synapse with a collateral ganglia called the celiac ganglion, which go out, and the postganglionic neurons that synapse here are going to innervate the stomach, the spleen, the liver, the pancreas, the small intestines, and the kidneys. Preganglionic fibers that synapse T9 through 12, the thoracic ganglia, are going to synapse in the celiac ganglia or the superior mesenteric ganglia, and the postganglionic fibers will reach out to the small intestines, the colon, and the kidneys. The lumbar ganglia, we remember that's L1 through L2, are going to synapse in the inferior mesenteric ganglion, and the target organs of the postganglionic fibers are going to be the rectum, the urinary bladder, and the reproductive organs. One difference that's the exception to the sympathetic chain ganglia or the collateral ganglia is sympathetic innervation of the adrenal medulla. Preganglionic fibers that innervate the adrenal medulla will not synapse in the sympathetic chain ganglia or collateral ganglia. These preganglionic fibers go directly to the adrenal medulla of the adrenal glands. If you think renal means kidneys, adrenal meaning a gland above the kidneys. So these preganglionic fibers of the sympathetic nervous system go directly to the adrenal medulla, the inner part of the kidneys, and release acetylcholine on chromaffin cells, which are modified postganglionic sympathetic neurons. When activated by the preganglionic fibers of the sympathetic nervous system, these chromaffin cells will release the hormones epinephrine or adrenaline and norepinephrine or noradrenaline. So question, preganglionic sympathetic fibers exit the spinal cord via the and pass through the to enter the all motor or efferent neurons are going to leave via the ventral or anterior horn or root. And if you remember, the sympathetic fibers are always going to run via the white rami communicantes and enter the sympathetic chain ganglia or trunk ganglia. So collectively, the white rami are called the white rami communicantes, and the gray rami are called the gray rami communicantes. Ventral root rami communicantes. So preganglionic fibers exit the spinal cord via the ventral root and pass through the rami communicantes, the white rami communicantes, to enter the sympathetic chain ganglia. True or false? The primary reason that the sympathetic division of the autonomic nervous system produces immediate responses within the body is based on the concept of divergence. This is true because if you remember, the sympathetic division has high neural divergence. The parasympathetic, if you were just to recall, the sympathetic division is often called the thoracolumbar division. In contrast, the parasympathetic division is often called the craniosacral division because it arises from the brain and brainstem and the sacral regions of the spinal cord. So the preganglionic fibers are going to arise from the brain and brainstem or the sacral regions of the spinal cord. These preganglionic fibers can then travel within certain cranial and sacral nerves. These are long preganglionic fibers whose axons will end or synapse in terminal ganglia that are near or in the target organs. So, because the terminal ganglia are in or near the target organs, the postganglionic fibers are going to be fairly short. So remember, in the sympathetic division, the preganglionic fibers are short and the postganglionic fibers are long. In the case of the parasympathetic division, the preganglionic fibers are long and the postganglionic fibers are short. Moreover, there's very little neural divergence. Four cranial nerves that are involved in parasympathetic innervation are the oculomotor nerve, which functions to narrow the pupil. This was that dual innervation example we were talking about earlier. The oculomotor nerve also focuses the lens. The facial nerve, cranial nerve number 7, that innervates tear, nasal, and salivary glands, and the glossopharyngeal nerve that innervates the parotid salivary glands, and the vagus nerve, which innervates many of the internal organs and blood vessels. Actually, not blood vessels, internal organs. If you remember, when we talked about the Biol reflex, we were involving both that Biol reflex with regards to the blood pressure in the heart involved both the glossopharyngeal nerve and the vagus nerve. The remaining parasympathetic fibers that are going to arise from the sacral nerves from S2 to S4 of the spinal cord. And they will form the pelvic splanchnic nerves that lead to the inferior hypogastric plexus, that then go out to the bladder and the reproductive organs, and the rectum, and part of the large intestines. So in comparisons of the sympathetic and the parasympathetic divisions, the origins for the central nervous system. In the central nervous system for the sympathetic division is the thoracolumbar spinal nerves. Remember, T1 through L2. For the parasympathetic, it's craniosacral, meaning those 4 cranial nerves and sacral nerves S2, S3, and S4. For the ganglia of the sympathetic nervous system, it's the paravertebral ganglia. Remember, that's also called the sympathetic chain ganglia, or the prevertebral, meaning the collateral ganglia anterior to it. The parasympathetic division, the preganglionic fibers are going to synapse with postganglionic fibers in terminal ganglia that are near or within the target organs. Relative fiber lengths for the sympathetic nervous system, the preganglionic fibers are short and the postganglionic fibers are long. In the parasympathetic nervous system, the preganglionic fibers are long and the postganglionic fibers are short. In the sympathetic nervous system, the neural divergence is extensive. In the parasympathetic nervous system, there's only minimal divergence. The sympathetic effects, neuronal effects, are often widespread in general. In the parasympathetic division, the effects are more specific and local. In addition to the sympathetic and the parasympathetic nervous system, we also most oftentimes include the enteric nervous system as part of the autonomic nervous system. The enteric nervous system is composed of approximately 100 million neurons found in the walls of the digestive tract or the gastrointestinal tract. The enteric nervous system does not arise from the brainstem or spinal cord. However, it is regulated by the parasympathetic and sympathetic nervous systems. Within the enteric nervous system, there's two plexuses or plexi. The myenteric plexus, which regulates gastrointestinal tract motility, how much things tend to move, the contraction and relaxation of the smooth muscle. And the submucosal plexus, which controls the secretions into the plexus. So how can autonomic neurons have contrasting effects on the same organs? One of the reasons is because the sympathetic and the parasympathetic fibers secrete different transmitters. Sympathetic tends to secrete norepinephrine. Parasympathetic fibers tend to secrete acetylcholine on their target organs. Also, the receptors on target organs can vary significantly, and target cells will respond differently to the same neurotransmitter depending on which receptor is present. So the receptor is what determines the response of the target cells. And different receptors for any given neurotransmitter can have different actions. So two different receptors for one neurotransmitter can result in different actions from the target cells. Despite the variety of possible chemicals that could be used to transmit chemical messages in the autonomic nervous system, only acetylcholine, norepinephrine, and epinephrine are used to any great degree. Synapses at which acetylcholine is used are termed to be cholinergic synapses. And neurons that secrete acetylcholine are called cholinergic neurons. Receptors that bind acetylcholine are described as either being nicotinic receptors or muscarinic receptors. Cholinergic neurons secrete acetylcholine. Receptors that bind acetylcholine are either nicotinic receptors or muscarinic receptors. I should also make another comment here. Actually, I'll wait on that comment once. Muscarinic receptors are the other type of acetylcholine binding receptors. And these receptors are found on all cardiac muscle, smooth muscle, and gland cells. These different receptors, these muscarinic receptors, can have excitatory or inhibitory effects due to the different classes of muscarinic receptors. So the plasma membrane of all effectors that are innervated by parasympathetic postganglionic axons have muscarinic receptors present. Plasma membranes of cells of most sweat glands innervated by sympathetic postganglionic neurons have muscarinic receptors. Nicotinic receptors are always found at the end of autonomic nervous system postganglionic neurons within the adrenal medulla. And these are always excitatory when acetylcholine binds. One thing that I should mention here is, if you remember, when we talked in chapter 12 about neurotransmitters, nicotinic receptors bind acetylcholine, but they're called nicotinic receptors because they also bind nicotine. Remember how nicotine stimulates as mimics the effects of acetylcholine binding. So the reason these are named nicotinic receptors is because nicotine also binds these receptors and mimics the effects of acetylcholine. These nicotinic receptors are also found at neuromuscular junctions with skeletal muscle, and are always excitatory when acetylcholine binds. Adrenergic neurons are going to form synapses where norepinephrine or epinephrine are secreted. And these synapses are called adrenergic synapses. So adrenergic receptors are going to bind norepinephrine or epinephrine. This right here would be an example of an adrenergic synapse. Adrenergic synapses are found at the end of nearly all sympathetic postganglionic neurons. We have two different classes of adrenergic receptors. We have alpha adrenergic receptors, and we have beta adrenergic receptors. Alpha 1 receptors are always excitatory, results in an excitatory response. Alpha 2 receptors are usually inhibitory. Beta adrenergic receptors, we have beta 1 and beta 2 receptors, and there can be different, we can have both excitatory and inhibitory effects related to both beta 1 and beta 2. So they can't be classified as being usually excitatory or usually inhibitory. It depends on the given receptor on the given cell. Can't classify them as being usually excitatory or usually inhibitory. So what comparison of locations of cholinergic and adrenergic fibers in the autonomic nervous system. Preganglionic fibers of the sympathetic nervous system are always cholinergic, so they always secrete acetylcholine. Postganglionic fibers of the sympathetic nervous system can be mostly adrenergic, meaning they secrete norepinephrine or epinephrine. But there are a few postganglionic fibers of the sympathetic nervous system that secrete acetylcholine. Of the parasympathetic nervous system, all preganglionic fibers secrete acetylcholine. In the postganglionic fibers, all postganglionic fibers of the parasympathetic nervous system also secrete acetylcholine. Another additional term that we need to describe when we talk about the autonomic nervous system is the idea of autonomic tone. And you remember we talked about dual innervation. What this is that there is a normal background rate of innervation by both of the subdivisions of the autonomic nervous system. Background rate of activation that represents a balance between the two systems. Where the rates of activation of one subdivision versus the other subdivision can change based on the body's needs. So dual innervation, where there's always a background level of innervation activity. And the given balance, which system is more involved in innervation of a particular organ or less involved in the innervation of that organ, can change from one division to the next. The other division, parasympathetic versus the parasympathetic, depending on what is needed at that given time. A sympathetic tone tends to keep most blood vessels partially constricted and maintains the blood pressure. The parasympathetic tone maintains the smooth muscle tone in the intestines and holds down a resting heart rate. So this changing balance between the two activities of the two subdivisions of the autonomic nervous system can be described further in an example of the heart rate. The parasympathetic nervous system tends to decrease the intrinsic heart rate. The heart is under an autorhythmic where it sets its own rate. However, the parasympathetic frequently decreases the autorhythmicity heart rate of the heart and lowers it because the parasympathetic nervous system tends to decrease that intrinsic heart rate. So the parasympathetic nervous system tends to reduce the normal autorhythmicity rate of the heart. While the sympathetic nervous system, when in the case of finding a bear chasing you down a trail in the mountains, like we talked about at the very beginning of this chapter, works to increase that heart rate and increase that intrinsic rate of the heart. Influences on the autonomic nervous system. The autonomic nervous system is regulated by feedback loops between the spinal cord and the brainstem, with additional input from the cerebral cortex, the limbic system, and overall oversight by the hypothalamus. So we have these feedback loops between the hypothalamus and the cerebral cortex and the limbic system that regulate feedback loops between the brainstem and the spinal cord when influencing the activities of the autonomic nervous system. Autonomic nervous system regulated by feedback between the spinal cord and the brainstem. With overregulation of the hypothalamus, which receives input from the cerebral cortex and limbic system. May we repeat that one more time? So the autonomic nervous system is regulated by feedback loops between the spinal cord and brainstem, And these feedback loops are regulated by the hypothalamus, which receives input from the cerebral cortex and limbic system. We talked a lot about dual innervation and this autonomic tone, where systems are constantly in a balance of innervation activity between the sympathetic and the parasympathetic nervous system. Changing balances. One system that is not innervated by dual innervation of the autonomic nervous system are the blood vessels. The blood vessel diameter is only controlled by the sympathetic nervous system. There is no parasympathetic innervation of blood vessel diameter. So vasoconstriction and vasodilation are both under the control of only the sympathetic nervous system. If you increase sympathetic nervous system firing, you get increased smooth muscle contraction, which results in increased vasoconstriction, reduced diameter. If the sympathetic nervous system firing goes down, the smooth muscle contraction goes down, and vasodilation, or increased diameter of the blood vessels, goes up. As part of this chapter is talking about drugs and their effects on the autonomic nervous system and effects in general on proteins and cells and enzymes and carrier proteins. Most drugs are targeted to our own human cells. Generally, drugs will act by binding to proteins like ion channels, enzymes, receptors, or carrier proteins, and either working to activate those receptors or block the activities of those receptors. So an endogenous normal receptor, like a regular neurotransmitter, binds to a receptor and produces an effect. An agonist drug can have an active site, an agonist drug can bind to the active sites of the receptor of a given neurotransmitter and produce the same effect as the natural neurotransmitter would. That is a drug agonist that mimics the effects of the neurotransmitter, binds to the normal neurotransmitter's receptor, and causes the same effect as the neurotransmitter would, the normal everyday neurotransmitter would, naturally occurring neurotransmitter. An antagonist drug will bind to a receptor of a given neurotransmitter. However, it will inhibit the effect of the receptor binding. So rather, it all it's doing, this antagonist drug, all it's doing is binding to the receptor and blocking the neurotransmitter from being able to bind to that receptor and cause the effect of the neurotransmitter. So all it's doing is preventing the receptor from binding, and therefore we have no effects. So agonist drug mimics the effects of the neurotransmitter. Antagonist drug blocks the effects of the neurotransmitter. Other types of drugs, for example, antibiotics, are targeted at the cells of pathogens that have invaded the body rather than our own cells. Monoamine oxidase is an enzyme that breaks down monoamines. And if you remember from chapter 12 when we talked about neurotransmitters, monoamines are a class of neurotransmitters. So this monoamine oxidase breaks down neurotransmitters like norepinephrine, epinephrine, dopamine, serotonin, and histamine. There are certain drugs that are designed to be monoamine oxidase inhibitors. So they inhibit this enzyme, and as a result, they slow down the breakdown of monoamine neurotransmitters. If you remember the three ways that neurotransmitters are removed from the synaptic cleft, as they can be reabsorbed by the axon terminal of the presynaptic cell. They can be broken down and then reabsorbed, which is what the monoamine oxidase does, or the neurotransmitter can just diffuse out of the synaptic cleft. What monoamine oxidase inhibitors are going to do then is they are going to slow down the breakdown of the neurotransmitter, thus keeping more neurotransmitter around in the synaptic cleft a little longer. So for example, Prozac, aka fluoxetine, is the generic name of Prozac, slows down the monoamine oxidase breakdown of serotonin, thus keeping serotonin around longer in the synaptic cleft. Levodopa works as a monoamine inhibitor to slow down the breakdown of dopamine, which, if you remember talking about the central nervous system, is the major part of the problem of Parkinson's disease. So it slows down the breakdown of dopamine, keeping dopamine around longer. So it sticks around longer and continue to have its effects rather than not being around when it's needed. Why is abuse of amphetamine so prevalent? Amphetamines chemically resemble monoamines. So amphetamine has a similar structure to dopamine. Methamphetamine, a derivative amphetamine, has a similar structure to epinephrine. Mescaline has a similar structure to norepinephrine. Because of their similar structures to these monoamines that are associated with elevated moods, therefore being drug agonists, the methamphetamines can inhibit monoamine uptake, promote their release, or inhibit monoamine oxidase activity. They can bind to the receptors, inhibit their breakdown, or promote more of their release of monoamines. Having the same effect, therefore these methamphetamines have the same effect or similar effects to the natural occurring monoamines that are associated with elevated moods and happier, more energized moods. So the mechanism of action of the methamphetamines is that they work as drug agonists, enhancing the activities of these monoamines involved in elevating an individual's mood. You remember one of the adrenergic subclasses of receptors were beta receptors. Beta blockers are used to reduce the stress on the heart. They enter the bloodstream via the GI tract, gastrointestinal tract, and they prevent norepinephrine or noradrenaline from binding to beta adrenergic receptors of the heart cells and vessels of blood vessels, walls of blood vessels. As a result, when the sympathetic nervous system stimulates the release of norepinephrine, the norepinephrine, even though it's present in the synaptic cleft, the beta blockers are bound to the norepinephrine adrenergic receptors, thus preventing the norepinephrine from binding and increasing the heart rate. Because that's what the sympathetic nervous system does when it secretes norepinephrine. So it blocks the effects of norepinephrine. Thus, even with the sympathetic nervous system firing to increase the heart rate will not increase because there's no receptors for norepinephrine to bind to. The result is that there's a decreased heart rate and as a result decreased cardiac output, which is the amount of blood that the heart pumps per minute. One last drug that many people including myself, struggle with an addiction to is caffeine. Why is it so addictive? Well, caffeine mimics adenosine, which is an excitatory neuromodulator. The natural binding of adenosine results in increased acetylcholine secretion from cholinergic neurons, leading to vasoconstriction. Caffeine is a drug antagonist that binds to the receptors that adenosine binds to and blocks those receptors. So inhibits adenosine from being able to bind to its receptor, which lowers the neuron's ability to secrete acetylcholine. Which decreases acetylcholine secretion. And the result is that rather than adenosine being able to cause vasoconstriction, when adenosine's binding is inhibited, can't bind its receptor, the result is that there's increased vasodilation instead of vasoconstriction when caffeine is present.